Hello and welcome to the poster presentation Basic4CVis, an R-Shiny app for 4C-seq quality control and interaction visualization. First, a short background for 4C-seq analysis in general. 4C-seq is short for circular chromosome conformation capture with sequencing. In a typical 4C-seq experiment, we have one main point of interest, the so-called viewpoint, and different areas with different resolutions. First, the near-cis area is close to the viewpoint and has typically good resolution. Then there's a far-cis area, the area on the same chromosome, where the resolution is a little bit worse. And then the trans interactions on other chromosomes, where we typically find no good resolution in total. The different levels of resolution possible make it necessary to think about different interaction visualization strategies to sort of capture these differences in resolution. And Basic4CVis, the R-Shiny app, is an extension of Basic4C-seq, an old package, where there are different visualization strategies implemented, especially for the viewpoint area with its rich fragment pool and data, and also some strategies for the far-cis and trans regions. So Basic4CVis, what input do we need? First, Basic4CVis can import different kinds of interactions. So basically all kind of interval data. We just need a chromosome start and end position, and perhaps also a score if possible. And we also accept separate upload for cis and trans interactions because it can be useful to vary it some input parameters for these interaction lists, and then we can just use different lists and upload them separately. Basic4CVis needs also a visual fragment library as input for the near-cis visualizations. This is similar to the visual fragment libraries used by 4C-seq, by Basic4C-seq, so the other R package. And there the input is basically the fragment data with the additional sample information. And there we can either use one single sample, then we have only the columns of chromosome start and end position and reads, or we can also add multiple additional samples. This is really just dependent on how many samples you have, and then you can just add them in different columns and use them also for some overlap analyses. The whole Shiny app looks like this. We have basically left some input options and upload options for the fragment library. This is basic for the near-cis visualizations. Then we can enter some viewpoint information. So in this case, I've pre-accessed this with, or preloaded this with chromosome 10 and some position here, and the species mouse. This is only relevant for the far-cis and trans visualizations when we do some circus plots and have the matching ideogram data. And we can also control the width of the near-cis plot here with this length in base pairs. On the right side, you can see the logging function where we can see what we have uploaded for now. So in this case, we have uploaded a virtual fragment library. We have uploaded cis interactions. We have tried to upload trans interactions, but gotten an error because we took the wrong file and then uploaded the correct trans interactions here. So there are checks in place to make sure that all the needed columns are really in fact in place. And the other windows here contain the plots, which I'm going to show on the next slides. The statistics and quality control tab here contains some, well, mostly length and score distributions if applicable. So if you've got the cis interactions and they contain a score, we can check how the score is distributed over all the samples, or we can check the length interaction. So 4C-seq has typically lots of, well, zero scores here if you take the whole fragment. So if you check the fragment library, this is probably more enriched on the lower scale here on the fragments with a lower score. But it can be interesting if you check a larger set of, let's say, cis interactions around the viewpoint or trans interactions, and you really want to have some distributions here. The more interesting plots are the near-cis interactions. Here we offer the usual plots of profile and heat map functions that Basic4C-seq also implemented. So this is really just well, additionally here with a peak visualization. So if we upload some cis interactions which are located in the near-cis region here, then we can just show these kind of peaks with some rectangles here. And as I said, we can plot multiple samples next to each other. Then the app just shows them partly, so starting with sample 1 and then under it sample 2 and 3, as dependent on how many samples are there. For the needs visualization, if there are multiple samples, we also offer an overlap function. This is calculated automatically. And here in this example, we have one peak that's obviously overlapping. So the overlap sample or the overlap plots for sample 1 and 2 will show this overlap in red. So as a different plot. And this will be the same for all the other samples if there are multiple samples. Then we have the 4C visualization. There we have chosen a so-called triangle plot, which basically only shows the viewpoint here as a spike. And then depending on whether there's a score or not, we also plot the score here on the y-axis. And then we just visualize a triangle reaching out to the interval as specified in the interaction. The same visualization technique is used for overlaps. So if there are sample groups and they have an overlapping interaction region, this is also visualized just with triangles. Circus plots are also available for the 4C visualization. There we just pick the relevant chromosome and do the standard circle plot. So starting on the area where the viewpoint is, we have these bands that reach to the intervals which are specified as an interaction. For the trans visualization, it's basically the same idea. We are just picking all the chromosomes here, and then we are visualizing basically over the whole chromosome, whatever they're specified as trans interaction intervals here. It's also available per sample or as an overlap plot for the whole sample group. So if there are multiple samples, then we have just the overlap calculation, and then only the overlaps are visualized in a special overlap plot here. If you want to read more on 4C-seq in general, I can recommend the Van der Werken papers from 2012, and probably also the original Basic4C-seq application note here. But it's a bioconductor package, so you can also check out the vignette. And for Shiny and R in general, it's of course a Shiny package, Shiny Web Application Framework for R, and the R core team publication here for the language and environment for statistical computing. If you have any questions or want to get in contact, my email address is here. And otherwise, I can only say thank you for your attention and see you at the conference.